Thank you for inviting me to give this talk on the molecular subtyping of esophageal cancer. In fact, I focus my talk on some slides that have been already shown by the previous, speaker. And I tried to divide the talk in, there is some difference between esophageal adenocarcinoma and esophageal squamous cell carcinoma. There is some difference between esophageal adenocarcinoma and gastric carcinoma, and what are the therapeutic implications of this difference. So this first slide summarizes a study published in the Nature recently, showing the let's say the omic data from more than 500 esosquamous cell carcinoma, adenocarcinoma of the esophagus, and gastric adenocarcinoma. You have the detail of the different type of tumors that have been studied, and as you can see, there is approximately 72 esophageal adenocarcinoma studied and 90 esophageal squamous cell carcinoma. The remaining tumor was, let's say, stomach adenocarcinoma. So the first thing, the first result presented by the author of this large study is they compare head and neck squamous cell carcinoma to esophageal carcinoma and gastric adenocarcinoma. What they observed, so they classify the tumor according different omic, the methylation of the DNA, microRNA, gene expression, copy number variation, and microRNA. And what you can observe from the place you are is that esophageal carcinoma can be divided in two groups. One group corresponds to head and neck, squamous cell carcinoma, and the second group corresponds to the adenocarcinoma of gastric adenocarcinoma. And these, in fact, seems that esophageal adenocarcinoma are closer to gastric cancers than to esophageal squamous cell carcinoma, which is more close to head and neck carcinoma. And if you apply the same classification based on the 4 different omic on esophageal squamous cell carcinoma and head and neck, and esophageal adenocarcinoma, in fact, you perfectly match the histological classification by these 3 omic. So you have strong difference between the two groups, and maybe we don't need pathologists to classify the esophageal carcinoma between squamous and adenocarcinoma. And if you compare now the gene expression and try to define the pathways that are differently altered between the two types of tumors, squamous and adenocarcinoma of the esophageal, in fact, what you observed, in fact, in adenocarcinoma of the esophagus, you have an upregulation of FOXA and OFF6 pathway, which regulate, in fact, E-cadherin. On the other hand, you have in esophageal squamous cell carcinoma, you have an upregulation of Wnt, CDKN, and P63, which, and this later one, is essential for the squamous cell carcinoma differentiation. If we go back here on the copy number, there is no, at your stage, probably you don't see any difference between the two types of tumors. Maybe there is more blue line here and maybe more red, but it's not clear from this at this stage. If we now characterize the allelic loss and the gain and loss of chromosome, and if you divide in 4 groups, and you put here the squamous cell carcinoma, here the adenocarcinoma of the esophageal, here the gastric adenocarcinoma with a chromosomal instability, and here the gastric cancer with non-chromosomal instability, You have a huge difference between these three types and these types, of course, because there is no chromosomal instability, and there is so no gain, no loss of chromosome, or very few of these. On the contrary, you have a lot of gain and losses, as you can see here, for these three types of cancer. And there is few difference, in fact. And the difference is only on this gray zone. Statistical difference is only on this gray zone. And this gray zone is due to difference between squamous cell carcinoma and adenocarcinoma of the esophageal or gastric adenocarcinoma. So the difference here, you have more frequent loss of chromosome 3p and more gain of long arm of chromosome 3. And you have here more gain in chromosome 14 in the squamous cell carcinoma, in the long arm of chromosome 14, as compared to the other type. And there is also some difference in chromosome 11 and 12. So once again, esophageal adenocarcinoma of the esophageals are closer to gastric cancer than to squamous cell carcinoma of the esophagus. Is there any difference in terms of mutation load between these different types of tumor? Here you have the difference between squamous cell carcinoma of the esophagus and adenocarcinoma of the esophagus. And in fact, esophageal adenocarcinoma have quite a large amount of mutation as compared to the squamous cell carcinoma. And for example, based in this series published on the ECGG, in fact, you have 1 log of difference in terms of mutation per megabase between squamous cell and adenocarcinoma. And as you can see, adenocarcinoma is just behind lung cancer and melanoma, which are the tumor with the most mutation load. Okay. Now I compile on my computer 210 cases of carcinoma of the junction from these different series published on TCGA, 2 on TCGA and 1 on by the Broad Institute. And this is the mutation load here. And this is approximately 2,000. The most important is 2,000, more than 2,000, approximately 2,000 mutation. And there is here no mutation or quasi no mutation. So you have a large difference between the different tumors, the 210 tumors. And as you can see here, we observed in those with on the patient that have the most number of mutation, the most large number of mutation, we observed 3 MSI positive patient, which is far less from what we can observe from gastric adenocarcinoma in terms of prevalence of MSI, because these represent approximately 1 or 2% of adenocarcinoma of the junction. And here is the different mutations, the most frequent mutation of the gene consensus. And what we observed, you can observe very frequent P53 mutation, which is associated with this chromosomal instability, as mentioned previously, and inactivation of CDKN2A by homozygous deletion and by mutation. You have an amplification of ARBB2 in 24% of the cases, inactivation of SMET4 in 20% of the cases, amplification of MYC in 20% of the cases, amplification of VGFA in 20% of the cases, amplification of cyclin E1 also, and you have an also an amplification of cyclin D3, an amplification of KRAS, which is present in 12% of the cases, and amplification of 10% in EGFR. So if we can now to group these alterations, because you have here the different type of tumors, and you can observe that you have a co-amplification of some genes and you can also compare the activation of the different pathway according to the different type of tumors. And what is shown in this paper that, for example, the MAP kinase pathway, which can be targeted, is activated in approximately 60% of the squamous cell carcinoma and 76% of adenocarcinoma of the esophagus. You have similar activation of cell cycle in the two types of tumors. 90%, I think it's 90, I'm sorry, I can see, it's probably 90%. I can see on my computer also, so it's 96%. What is interesting is that the way the genes are activated or inactivated can be different between the two types of tumors. Here you have an activation of, the inactivation of CDKN2A is in fact due to silencing by hypermethylation in adenocarcinoma or by deletion in squamous cell carcinoma. And in fact, you have approximately the same percentage of inactivation of the two genes, but the mechanism of the inactivation are different between the two types of genes, the two types of tumors. As you can see also in the pathway here, you have an activation in 38 and 50% in the, of the, let's say, cell proliferation pathway. You have also cell differentiation that are activated in the similar frequency, but if you focus on the way these pathways are activated, there is some difference between squamous cell carcinoma and adenocarcinoma. That's also true for chromatin remodeling with mainly ARD1 and SMARC4 in adenocarcinoma and lysine methyltransferase in squamous cell carcinoma. So the pathway activated at similar rates, but the gene targeted at different, and mechanism of activation and inactivation can be different between the two types of tumors. If we go back to this diagram and the different type of tumors that have been shown in this paper, as mentioned by the previous speaker, there is different prevalence in chromosomal instability for gastric, for carcinoma of the junction as compared to the body of the stomach, for examples. And there is more prevalent SYN phenotype in esophageal adenocarcinoma than the other stomach type of tumors with less, and you can see there is only 10 with a gastric stable tumors. There is no or very few MSI tumors, and there is no EBV tumor in the gastric junction. So this is a representation in the different type of gastric esophageal junction carcinoma. This one is those that are on the side of esophageal. These are indeterminate in the medium. This is a carcinoma of the junction from the gastric part of the junction, and this is for fundus and that for pylorus. And you can see the different prevalence of the 4 different phenotype or genotype of the gastric carcinoma with a very homogeneous type of tumors in the esophageal part of the junction as compared to the other part, and an increased number of MSI tumors from the top to the bottom, and also an increased number of stable tumors from the top to the bottom. If we go back to methylation, we can observe some difference between the different type of junction tumors, as the prevalence of methylation are higher in the hypermethylation is more prevalent in the upper part of esophageal esophageal part of the gastric junction, as you can observe here, with a more prevalent of C1 and C2 cluster, which are the cluster of hypermethylation genes. just to mention these table, you have different genes and different alteration of these genes, the frequency of the alteration in terms of silencing or mutation, in blue, inactivating mutation, or in red, activating mutation or amplification of the genes. And what is observed here, for example, you have a gradient of alteration from the esophageal part of the junction to the antral part of gastric carcinoma, with low frequency of APC, for example, that increase from up to bottom. This is the same thing, for example, for SMAT4, you have a decrease from the top to the bottom. You have an increase in terms of, you have more silencing by hypermethylation in the upper part of the junction as the lower part. You have an increase of, you have a decrease from the bottom to the top to the bottom for inactivation of CDKN2. You have similar rate for BB2 activation, but you have, for example, more amplification activated VEGF-A in the upper part of the junction, for example. So there is some difference that could lead to different approach in the treatment of this tumor. This is the occurrence of the different amplification on different genes, cell cycle genes, RAS, or say MAP kinase genes, or other type of genes, and you can see that there is some co-amplification of different genes, and this amplification can be targeted by different therapeutic trials. So the need of the characterization is probably an essential point to adapt the treatment. And what has this observed in another publication in Nature Genetics recently, 2 years ago now? In fact, they studied 129 tumor of the junction. And in fact, here you have the representation of the deletion and the activation by amplification or gain or loss of chromosome, loss in blue, red, gain, dark red, amplification. And what you can observe, in fact, that from one tumor, which is one line, you have co-amplification in tyrosine kinase receptor or downstream or in the downstream pathway of this tyrosine kinase receptor. So, in fact, this could explain in some way the failure of some targeted therapies that have been used in the gastric cancer, because if you have several genes that are amplified in the same tumors, and if you target only one gene, of course, there is some way to escape to this treatment. And if this summarizes, in fact, that in fact you have co-amplification in the tumor of the junction in approximately 83% of the cases. That's the vast majority of the cases of the gastric cancer of the junction have in fact co-amplification in different receptor kinase. And if you add amplification downstream, you have 85% of the cases with at least one gene amplified. Or these amplification or gain of chromosome also translate in terms of gene expression in most of the cases. You have here some tyrosine kinase receptor or MAP kinase protein. You can see that there is some significant difference between these two groups of tumors, which can be which can translate, in fact, the role of the amplification in terms of selection of tumors. So you can also, based on these results, you have some cell lines that reproduce what is observed in human tumors, and they take some examples to the combination of different different targeted therapy to be more efficient. Here is the example of 5 cell lines. And for example, these cell lines have a co-amplification of ERBB2 to MET in the same cell line, so you have the 2 genes. So you can treat by lapatinib, by crizotinib, and you have these area under the curve here. And you see synergistic effect of lapatinib and crizotinib in this cell line by inhibiting the two targets, in fact, by combining the two targets. So you have also several examples showing a potential effect to combine two targeted therapy in this type of tumors. I would like to finish the talk by the demonstration that we can go further in the classification. Here you have an example of signature, what we call a mutational signature. If you characterize a mutation in one tumor, you can characterize 6 types of mutation: C2A, C2G, C2T, T2A, T2C, and T2G. But you can also consider the base before the mutation, the base before the mutation, or the base after the mutation. So you have now 96 possibilities, which are represented here by these different positions. So you have, probably you recognize this type of tumors. This is a tumors, this is a melanoma. Because this pattern of distribution of mutation is typical to tumors that have been exposed or is due to UV exposition. So this is a pattern which can be recognized as a pattern of melanoma. So we don't need any more of pathologists. So now there is 30 different types of signatures that have been recognized, at least, by sequencing, and we can recognize different patterns. This is due to deamination of 5-methylcytosine, and it's mainly due to the age. There is no risk factor, specific risk factor. This signature is due to the exposition to alkylating agents. This signature is due to aristocolic acid, this to aflatoxin, for example, this to deficient mismatch repair. These signatures are observed in MSI tumor, for example, and this signature is DNA damage repair deficiency, for example, and is mainly due to deficiency in BRCA1, BRCA2 genes. So they applied this type of analysis to gastric adenocarcinoma, and more particular of the junction, and they classified the tumor according to the presence of these different signatures. And what they observed, in fact, they are able to classify the 129 gastric junction cancer in 3 groups. One is recognized as a signature 3, which correlate with the homologous recombination deficiency due to BRCA deficiency. This is due mainly to a signature S18 or S1, and is a C280 in a CGA-TC context. And you have the last group, which is due to a signature. The main signature is a signature 17, which has been divided into groups, and as you can see, is mainly T2G in a CTT context, which is represented by this peak here. And as you can see, the number of mutations of this different type of tumors is not homogeneous, and this subgroup with this signature is in fact accumulate mutational. And this type of tumor have a high mutational burden, and probably due to the exposition to acid, gastric acid reflux. From this classification, they calculate the neoantigen presentation, and as you can see, the mutagenic type, which is this one with the signature 17, have in fact more neoantigen presentation, a number of expressed neoantigen significantly different from the other type, and also an infiltration by CD8 cells more prevalent. So probably, once again, we can suppose that this type of tumor could be, for example, treated by immune checkpoint inhibitors. Sorry. This is a repartition of the different signature in 3 different cell lines, and they applied some drug. For example, in these cell lines with a signature of DNA DNA damage repair impaired. By the combination, you obtain some sensitivity by a combination of olaparib, a PARP inhibitor, associated with inhibitor of topoisomerase, which is an enzyme necessary for replication. This type with a mutagenic signature could also respond to checkpoint inhibitor, G2M checkpoint inhibitor, WEE1 or CHEK1 or 2 inhibitors, and this has been demonstrated in cell line. So there is different combinations that could be applied. So to summarize what's of the different subtypes, we can now divide this subgroup of SYN from the gastrointestinal junction, for example, by 3 types, in fact, mutagenic, DNA damage repair impaired, and C2A and T mutation, C to A or T mutation dominant, which can be treated from different ways. This type of gastroesophageal junction, 29% of the cases could be treated by conventional chemotherapy and also probably by tailored ERBB2 MET inhibition, because you have an enrichment of ERBB2 MET amplification in this subgroup of tumors. You have those with DNA damage repair impaired, 18% of the cases can be treated by combining, let's say, damaging agent, DNA damaging agent with PARP inhibitors, and this one with a mutagenic profile with T2G mutation in the CGT context. In fact, have a highest mutational burden, a highest neoantigen load, and probably can answer to immunotherapy, inhibitor of checkpoint, or inhibitor of cell cycle. So in conclusion, adenocarcinoma of the esophagus is closer to gastric carcinoma than to esophageal squamous cell carcinoma from a molecular point of view. As compared to gastric cancer, there is fewer MSI tumors and virtually no ABV tumors in the tumor of the junction. There is some difference between carcinoma from upper and lower part of the junction, mainly in methylation pattern. And there are also slightly different in the frequency of cancer gene altered, with a continuous gradient from upper part of the gastric junction and to the lower part of the junction. And there is mutational pattern which are clearly different in some cases from what is observed from gastric cancer. And probably we can use this mutational pattern to define some new targeted in cancer, in gastric cancer. Thank you.